Have you ever heard of low-T copper? So you search it on your manuals, like any DNA dissolving or any other manuals, you find low-T copper. So low-T is basically 10 millimolar trees and 0.1 millimolar trees. Okay. So you use that buffer for dissolving DNA, the extracted DNA, if you are extracting manually. If you are using a kit for DNA extraction, that low-T copper is already there in your kit. So, and for the dissolving of your primer, you maintain the low-T copper. So, can you able to tell me that what is the primer concentration? Whenever you are getting a primer from a company, so it is lyophilized. So, how you dissolve this primer? What is the concentration, stock concentration of this primer? 100 micromolar. So, any primer sheet having that information that how much amount of liquid you need to add to get it in a 100 micromolar concentration. Correct? So, you are dissolving in low-T copper in a 100 micromolar concentration. And from that 100 micromolar concentration, you need to create a stock of 10 micromolar concentration. So, that can be utilized for your PCR. Okay, for your PCR purpose. So, now, when you are going for PCR, suppose you are every day you are doing PCR and you are getting some primer timers. So, have you seen primer timers in many of your PCRs? So, primer timer comes when there is some excess primer and primer is actually self-complementary, self-annealing and self-complementary. So, this is the possibility. So, then what you can able to do, you can reduce your primer concentration. Keep it at 5 micromolar, no harm. If you are getting more primer timer, reduce the primer concentration in next reaction. Then you will actually get rid of the primer. So that's how actually you need to calibrate your PCR. So primer timer cannot be removed by any method other than gel extraction. Okay, primer timer will be there. So that's why you need to properly emphasize on your PCR product that there should not be no primer timer or very minimal primer timer. So what will be there in the primer timer? Suppose I will be coming to the troubleshooting answer. Suppose this is your sequencing reaction, sequencing data.
It's not working.
Okay.
Take this one. Take this one, sir. Yeah, this is good.
And if there is a primer timer, you will see that there is a peak like that, shorter peaks like that, around 50-60 base pair. You will be getting. So because of that, your sequencing reaction may fail. Okay. So that's why you need to avoid any primer timer in your PCR. So first of all, you need to check with this the salt and EDTA. It inhibits the DNA polymerase and preferential injection. So that's why when you are performing the cleanup method of your cycle sequencing, I will be discussing about the same. So you need to be very cautious about cleanup of your PCR products. Okay, cycle sequencing PCR products. So now this is very crucial. So when you are actually considering the template quantity, how much DNA quantity you should take as a cycle sequencing template DNA amount. So the optimal template quantity should be empirically determined due to variation in the template purity, method and measurement and the BigDye Terminator dilutions. So the thumb rule is that PCR product length, how many base pair divided by 50. That is the thumb rule of the total concentration of the DNA or the PCR product you should take. And the standard BigDye Terminator protocol tells about that PCR product length is around 500 to 1000, so you should take not more than 20 nanogram. So as a whole, for sequencing reaction, it is not a quantitative reaction, it is a qualitative reaction. But again, when you are taking the PCR product, you need to consider about the concentration. More concentration will be inhibiting your cycle sequencing reaction, and it will be giving you suboptimal results as well. So quantify the data. So quantify by any method like 26020, any qubit or any other method you just quantify the data. Okay, or even when you are running in gel, you can gel estimate also. So gel estimate it and write it down on your copy and give it to the central facility that yes, this is my gel picture and this is my quantity of the PCR product. So that they could understand that how much amount of DNA should be taken from your PCR product to perform a cycle sequencing reaction. Okay. is the thumb rule of your Terminator protocol. So now I told you about the DNA quality and quantity. So agar-usual electrophoresis that reveals the presence of contaminating DNA and the RNA. If there is any contaminating DNA, you will get extra bands. And protein you can't able to see. Purified DNA runs as a single band. So you will get a single band. Estimate the DNA amount using ladder with the known concentration. That also you can able to do. So then you can able to do a spectrophotometric method 260 to 80. So you can able to get the, it indicates the presence of protein contamination also. So 230 also you can able to get the salt contamination as well. So 260 to 80 ratio and 230 to 60 ratio you can able to provide to the laboratory so that they could understand about the quality of the DNA. Every time you are providing it to your laboratory, you provide this information to your laboratory. Okay. So avoid protein detergent, I told DART and salt and EDTA buffers, already I told it. So now what affect with this polymerase activity? So suppose there is a sodium chloride present or any, so for example it is sodium chloride, so any salt present in your reaction. So first 100 base pair data will be very good, after that you will be getting a very flat data. While if there is no salt, so then you will be getting the sequence and then after that you will be getting also a good sequence. So that is the comparison of PGM control in presence of 40 millimolar sodium chloride or water and effect of salt on the polymerase activity. So now coming to the primer designing. So primer should not be more than 25 millinucleotides. Ideally it should be 70 to 25 millinucleotides with an average TM of 55 to 65 pp. So you need to maintain the primer around that. And if you are working with humans or any other species, so you go to the primer designer software in Thermo Fisher website, you will get many standard sequencing primers. So these are all pre-designed primers. Okay, primer designer tool. So that is a free tool available. It is a web-based tool. No software need to be installed. Just search for with your gene name or your mutation name or RSID or any other particular nomenclature. You will find out these primer sequences available from the website. Okay. If you don't get, you actually rely on the publications available. Right. So general rule for annealing temperature is that temperature of annealing is basically less than 5 degrees of the temperature of melting. And how you calculate the temperature of melting? You know that 80 into 2 plus GC into 4. So that is the 2 plus 4 rule basically to calculate the temperature.
I don't know whether you can or not, but that's a storage of primer is very crucial thing. Right?
I'm coming.
Okay.
I'm coming. So that will be there in my report. Yes. So avoid primer dimer when you are designing your primers. Avoid hairpins in your primer and also loop formation, secondary structure and secondary binding sites. So I told you that even if you have the primer at a secondary binding site in between, so the gel extraction will not help you. So that's why avoid gel extraction even if there is a secondary product available. So we actually perform this PCR to avoid the secondary.
So I have a question. Like when we are designing the primers, we get all these parameters and whatever software we are designing it, dNTP or Gene Runner or whatever. Now we see that we have to make a decision. Like either we have to stick to the, like it's not a foolproof thing. Either we get the TM and everything in a correct position, or we get the primer dimer setting. So we need to balance it.
Balance it
out. Because one or two timers might be present somewhere, and the hairpins and secondary structure, the little bit amount is present.
You will not get an absolute change. So it is a balance between your primer binding. So that is the binding score as well as these hairpin structure.
That is we cannot completely avoid it because I have seen my designing primers that it's not completely avoidable. However, we get the PCR and everything single band and everything works fine later on.
Yes.
But so what is actually the threshold that we can really permit for designing the primer?
For primer designing, you can able to follow primer 3 or any other software, so which can guide you that what are the thresholds. So you can able to keep those thresholds.
Those things in mind while selecting the primer, which is.
And it will be very good if you have a pre-designed primers available in their publications also. Means if somebody has published and given the data, so then it will be a foolproof solution for you that it is a published primer and you can go ahead. If it is not published, then design accordingly. That
what are the parameters that we actually have to follow? Like some permissible amounts that is there.
So I told you that you should should actually stick with the less GC content, because more GC content will increase your TN. GC content.
But GC content also has to be a little bit like around 55
You can't avoid, because that is actually depending on your total product sequence. So anyway, you can able to adjust between
few base pairs here and there.
Few base pairs, and you can able to go with the base.
And also they have to give a certain amount of GC content so that it anneals properly.
Yes.
Otherwise, the annealing won't take place if that is not.
Right. And also after designing, so it is a in silico design. So then also when you are
there are chances
PCR,
that
so there is a chances that the PCR will fail, PCR will produce you multiple products and so on. So in that cases you can try with various commercially available tag polymerases like platinum tag, gold tag. So those are the tags available in the market, and you can try with those tag polymerases to get ideal PCR product.
So where do I like if I am designing a primer and I am trying to do a PCR? Where do I get the thresholds for all these hairpins and secondary
So
structures?
it will show you now whenever you are designing your primers, it will show
It shows.
that,
What is the permissible limit?
so that you can check with those softwares, that they will guide you. The softwares will guide you, because primer designing I can take another class for you, that what should be the threshold. So that I can able to guide you separately. But in the interest of time today, that I am giving you a scenario that what you need to avoid for your primer designing today. But yes, again, if you want to know about the primer designing, I will be happy to provide you all the information. So come back to me. I will be guiding you as much as possible. Okay. So and then the next part, what you asked for, primer storage. The resuspension, I already told you that lyophilized primer oligos resuspented in water. Water is good when you are doing it for a lesser time. So like within 6 months you will be utilizing your primer, no problem, you will do it in water. But for longer storage, you do it in tea water. It will be stabilizing more effectively with your primer. Otherwise, primer will be broken, some one base pair or two base pair, you may not know that. The primer quality will be degraded. So then N+1 effect will be there in your primer sequence. What is N+1 effect basically? So this is the primer.
Kept it there.
This is the primer, and suppose this base pair is actually cut off because of the storage and all. So then another primer actually came up in your total primer mixture, which is one base pair less. So then two primers will be there in your mixture that will actually go for the sequencing. So what you will be getting, that one peak, then another peak, then another peak, then another peak, like that. So that will be effect of your data in your sequence, because ultimately it is one base pair less than this primer, and due to the degradation you are getting this kind of data. So due to the primer degradation. Okay. So you dissolve it in your 1 millimolar tris and 0.1 millimolar EDTA. Left for 10 minutes, vortex and alicot. Always alicot your primer, because multiple tris thaw will actually degrade your affect your primers also. So properly store it and measure the quantity if required, but don't require, actually it is given that this much amount of primer is there, and dissolve it in that much of microliter of the water tris buffer. Then store undiluted stocks at -20 or -80 for long term storage. And working solution, that means the 10 millimolar, you keep it at 4 degrees. Do not freeze multiple times. So many times of freezing and thawing will actually affect your primers. So store it properly. And short term storage up to 1 month can be possible within 4 degrees Celsius. Okay. So store it according to your primer storage instruction. And calculation, I already asked you that how you are dissolving your primer to 100 micromolar. So it is basically 100 picomole per microliter stock concentration. So nanomole oligo into 10, that is the volume of buffer to add to the oligo. So example, you have a 9 nanomole of oligo and you need to make it 100 micromolar. So 9 nanomole oligo into 10 is 90 microliter of the buffer needed. So though I am telling it, so every primer document provides you these instructions. Okay. So how the primer storage quality affect? So you see that there is another peaks are coming. So this is due to the primer storage condition, and then another peaks are actually coming. So because of this primer degradation, so another extra signal is coming. resuspension, I already told resuspension in T10E.1. That means it is a low T. So it is a low T. Okay. So measured quantity is spectrophotometer. Storage, I already told. So primer dimer. So this picture I already shown you that the presence of primer dimer provide another template around up to 60-70 base pair, and it will actually damage your total sequence. So this is the total workflow: DNA extraction, amplification, cleanup, cycle sequencing cleanup, electrophoresis, and data analysis. So this part, the cycle sequencing and capillary electrophoresis, will be done with the kits provided to you. So it is called a cycle sequencing. So these are the workflow associated products. So DNA extraction and PCR amplification, I already told that you can able to utilize the AmpliTag Gold or any other platinum tag for your PCR amplification. So for PCR product cleanup to cycle sequencing cleanup, we do have solutions with the ExoSap available with us or any other provider. Cycle sequencing kits, the BigDye Terminator one, there are two kits available from Thermo. Mainly, one is version 1.1 and version 3.1. So whatever in your laboratory you are utilizing is the version 3.1. Version 3.1 provides you a longer bit sequence, and it is unanimously used for variety of sequences. So that's why version 3.1 is the more popular kits available in the market. And then you are cleaning up the cycle sequencing product either with the Xterminator or with ExoSap or Dyna bits or with the advanced precipitation method. So these are all the methods available. And then you are running it in the genetic analyzers and data analyzed with the data analysis software. So PCR reaction product must be cleaned up prior to sequencing. I told you that if you are not cleaning up properly, then the dye-terminators reactions will be there, and that will affect your PCR data, sequencing data. So sequencing template preparation, removal of PCR reactions, that is the primers. The beginning slide I already discussed it, that primer to avoid multiple sequence, and dNTP is to keep the optimal dNTP and ddNTP ratio, and salt to avoid inhibition of the sequencing enzyme, and non-specific product to avoid the sequencing artifacts. So this we need to remove. Yes.
So what is the normal BDD ratio of dNTPs and ddNTPs?
That is not discussed. So there are two types of kits I told you. One is 1.1 and one is 3.1. 1.1 having the more ddNTP ratio, so that it will be giving you a shorter product size. But at the beginning, you know that from your cycle sequencing product, at the beginning of 20-25 base pair, you are not getting good data. So it is because of the ratio of dNTP and ddNTP. So if you want good data at the beginning, use the 1.1 chemistry or the BigDye direct chemistry with the Enzymatic. So there are different chemistries available. Okay. So I told you that there are methods available: ExoSap, column purification method, jet purification method for your PCR product cleanup. So try to work with either with column or ExoSap method. Sorry. In the next, I will be telling about the PCR products. So optimize the PCR reaction. Quality of the PCR product is very crucial. So here you see that here there are two PCR products came, 1 and 2. Here there is a PCR product and this is the primer dimer, and here it is a single slide. Okay. So now I have a jet purification. Can be used for purification of PCR product, and it eliminates the non-specific PCR products and primer dimers also. But disadvantage is that variable and low yield, time consuming and UV leaking. That's what I told you. Okay. There are various commercially available kits available. Now with the column. So with the column you can, it is fast and reproducible, but the disadvantage is that it is relatively expensive and more time consuming, because you need to transfer it to column, wash it, and then elute it, and then utilize it for your sequencing kit. It takes time. So then the enzymatic purification, if there is only single band available. So just add your enzymes and treat it for 15 minutes at 37 degrees centigrade so that it will destroy all the primer and dNTPs, and then inactivate the enzyme, because inactivation is important. Otherwise, that enzyme also degrade your primers available in your cycle sequencing and also dNTPs in your cycle sequencing. So that's why it is also required to deactivate the enzymes as well. Any questions? Please, any questions? Any thoughts?
It is the ExoSap method, right?
It is the ExoSap. So I told you about all the methods like the gel purification, column purification, ExoSap, because I know that in various laboratories people follow various methods. So that's why I provided advantage, disadvantage. What will be the probability? I am not telling you that this is good or this is bad, but you need to understand that what should be effective for your own research. Okay. What will be the ideal method? And direct sequencing. So somebody I have seen in many laboratories that they are getting a huge amount of PCR product. So now they are diluting the PCR product in such a dilution that other dNTP will be diluted. So if you can establish that protocol well and good, but it is risky. It is risky because you are diluting it to 100 times. Then also some primers and dNTPs may present in your reaction. So if you can establish it is well and good, but it is risky. Somebody do that. So then coming to the ExoSap, what it is actually doing. So ExoSap is basically removing these primers and nucleotides, and you are getting a very good clean data after the ExoSap cleanup. I am not telling you about the cycle sequencing theory part, that how the ddNTPs is actually binding and all. So you know that. So what it is actually going through is annealing, extension and denaturation of 25 cycles of PCR. So that is the standard method. But if you know that your PCR product and primer concentration is high, and you are getting a very high amount of cycle sequencing product, so you may reduce 1 or 2 cycles. It is up to you. But in general, we try to follow these 25 cycles of PCR. That is the universal protocol people used to follow. Okay. And here you are utilizing the dNTP or ddNTP, both buffer, polymerase and template DNA, and also the primers together. So you are getting a product like primer plus 1 base pair, primer plus 2 base pair, and so on. So this is how the products are being generated. So then cycle sequencing reactions, you are getting a multiple products after the elongation of 25 cycles. So all the linear amplification of single stranded products with the dye label at the end. Okay. So because why it is one single dye label, why it is not multiple dye label? Can you able to tell me? So when you are getting a PCR cycle sequencing product, so every product is one strand with one dye label. Why it is?
Because the didioxynucleotide digestion at the end prevents
So we are utilizing the mixture of dNTP and ddNTP. When dNTPs is adding, so it is elongating. When ddNTPs is adding, it is stopping. And at the stop point, it is the dye. That's why we are getting a unique sequence with the dye. Okay. And for every sequence or every particular base, you are getting one product. So total, you are getting these chain termination sequences after the number of cycles. And then in the mixture, you have the sequencing products and also the ddNTPs available in your products. So in the next step, after the cycle sequencing, you need to remove these ddNTPs. Primers, it will not happen because primers will not have any dye. Okay. So unused dNTPs are having the dye. That you need to remove. So that's why cycle sequencing cleanup method utilizes like ethanol precipitation, like the exterminator, like the magnetic field-based cleanup, same. Though the first idea is to remove the ddNTPs. It will affect your DNA. Okay. So it will amplify and you will get the product, and then you need to separate it from the matrix. So then coming to the chemistries. So leveling of extension product. So BigDye Terminator chemistries are available. That is actually providing you the extension product labeled by fluorescent dyes, and at the 3' end the BigDye Xterminators want different dyes for each dNTPs is adding. So here is the question. So when you have only all ddNTPs available in your mixture, what will happen? At the first base it will stop. There will be no amplification. So you can't do sequencing reaction with only ddNTPs. Okay. So this is also having an application called the snapshot application or the single base extension application. That application is also there, but that is fragment. Last time I told about this. So then low, that means more dNTPs and more ddNTPs and less dNTPs. And then ratio of dNTP to ddNTP is high. That means more dNTP and less dNTP. And at the end there is no ddNTP. That means you will be getting products, but you will not get any sequence. All products will be of single size because there will be no chain termination. Okay, these are the 4 scenarios. Out of these 4 scenarios, 2 scenarios has been taken to consider for the development of sequencing reaction. So this low ratio is utilized in a version 1.1 chemistry and high ratio is utilized in the version 3.1 chemistry. Okay. Single base, mostly short extension products and more or longer extension product and no product. And in the next slide, the different kits are available. So this is the shorter extension product with the BigDye Terminator version 1.1 chemistry and longer extension product with the BigDye Terminator version 3.1 chemistry. So the different sequencing kit for different sequencing applications are being developed. So what are the applications? So like we have the BigDye direct kit, Terminator 1.1 and 3.1 kit. So all types of sequencing can be done with all the kits. Okay. So then high resolution close to sequencing primer if you want. So then you can able to use either direct or 1.1, not 3.1. M13 tail primer, M13 sequencing primer, you can able to use more effectively with the direct kit and there is no purification is required. So then universal tail primer other than M13 primer. So you can able to use the BigDye Terminator either 3.1 or 1.1. Long read always 3.1. Mixed base, BigDye direct, GC rich, all are same. Like it is providing a satisfactory data. So yes.
So how close that you can get the resolution to the sequencing primer with the BigDye direct and
BigDye direct is also higher ratio and BigDye Terminator 1 is the lower ratio. That means dNTP is low and ddNTP is high. Direct means it is a kit developed with the faster sequencing product solutions.
But the second point is high resolution close to the sequencing primer.
High resolution close to sequencing primer. Why? Because the BigDye direct kit utilizes an extra M13 primer. So that means it actually starts the PCR product little bit.
How close?
It is around 5-6 base pair of the PCR product. So extra 5-6 base pair is actually amplified and then ddNTP is added to that.
Okay.
And here it is direct primer you are using. It is M13 primer you are using and it is the direct primer you are using. For BigDye direct always you need to use the M13 primer. It is a universal primer. So that means it should have, your PCR product should have an overhang of M13 primer and with that you can able to sequence. So with any primer you can't do a BigDye direct sequencing. BigDye direct is a separate kit. It is utilized with the M13 primer overhang. Okay. But concentrate about these two kit solutions, the Terminator 3.1 and 1.1. So 1.1 you can able to get from 5-6 base pair you will be getting a good product. Why? Because I am telling you here. Because so when there is a high ratio of D, suppose this is your primer. So then your ratio of ddNTPs is very high. So the probability of generating another product with a dye tag at the primer end will be very high. Because more and more ddNTPs is utilized than dNTPs. So that the concentration of the product at the beginning will be very high. But since it is very high, so it is utilized more, so that the longer product possibilities will be less. So within 200 to 300 base pair it will be all utilized. So you will not get a very long product with the BigDye Terminator 1.1 kit. Version 3.1 you will get a longer product. As well as you are getting a closed sequence very close to primer.
Yeah, so in our lab we are using version 3.1.
Version 3.0 direct.
Yeah.
So 3.0 direct is 3.0. Yeah.
But 3.1, I think doesn't give that much
3.1, yeah, 3.1 at least 25 base pair you will be not getting a good data initially, because you are getting a very less product at the beginning. More and more dNTPs will be utilized because here the dNTP ratio is high. So then dNTPs is more utilized. So you will not get a very effective amount of fluorescence, so that you will get a peak over there. So that's why with the 1.1 you will get a closer DNA sequence closer to your primer. Okay. But anyway, when you are doing a sequencing, a longer sequencing, you concentrate with the 3.1. But yes, if you have a very short product size, 200 base pair, 300 base pair, I will provide, suggest you to use the BigDye version 1.1 for those sequences. Okay. So, BigDye Terminator kit contains the ready reaction mix. Ready reaction mix contains all the mixture provided for your sequencing, that is dNTP, ddNTP, your enzyme, your buffer, everything except your primer and DNA. All the products are inside. Okay. So now it is coming in a concentration of 2.5x. So when you get a sequencing reaction kit, you will get as it is a 100 reaction kit. But 100 reaction kit can work 400 to 800 reaction because you can dilute it. So more the dilution, you will be getting the lesser peak heights, but you will get the sequence because it is not a quantitative data. Okay, people used to dilute it and we provide also 5x dilution buffer along with the kit as well. Okay, so based on your sequence, because we do not know your sequence may be, your PCR product may be very, very less. So in that case, you need to utilize more and more enzyme to amplify. So in that case, you need not to dilute it. Or again, you have a very good quality of the DNA and quantity of the DNA, you dilute it and go for sequencing. It is up to you that how much dilution you will be doing for your PCR. So that's why it is having a 2.5x concentration. Always run a controlled sequence along with your everyday sequencing. Suppose in a facility, in a central facility, you are running 20 total sequencing today. Along with 20, I have one positive control along with the PGM or PREF right now. So PREF is a vector and having the known DNA sequence that is inserted in the plasmid and it is given inside the kit. And along with the M13 primer, you amplify it. And we also provide the 5x sequencing buffer, shipped separately, and you need to store the sequencing buffer at 4 degrees, but all other ready reaction mix and control should be in -20. So my suggestion is that if you are everyday utilizing the sequencing RR mix, you keep it in aliquots. The RR mix should be keep it in aliquots, so that every day you need not to freeze thaw your RR mix. Okay, so that is also another suggestion for you. So these are the total kits, ddNTP and nucleotides and all other solutions are provided. So one ready reaction mix and then buffer and then the control DNA and the control primer. So these are the 4 tubes available with the sequencing kits. So already I told you that there are certain kits available. 3.1 is optimized for long reads. 1.1 is for challenging and short read sequencing, and the direct kit is combines post PCR cleanup and cycle sequencing into a single step, but requires the extra primer with the M13. So most popular long read for good for everything, challenging template for shorter reads, and huge time saver. So for diagnostic purposes, having the all the samples with the M13 primer, you can able to utilize with the direct kit. So this is the data of the Terminator kits between the version 1.1 and 3.1. So this is the heterozygous detection. So you see that version 1.1 having the heterozygous having the different peak heights, but version 3.1 heterozygous having the similar peak heights. So this is how when you are doing with the heterozygous analysis, you always use the 3.1 chemistry, not the 1.1 chemistry. So more homogeneous peak pattern and better identification of the mix basis. But when you are getting the data from the start sequence, so this is the start sequence of the 3.1 chemistry and this is the start sequence from the 1.1 chemistry. So if identification of the base of the heterogeneous is within the 50 bases is required, run with the 1.1 gig chemistry. So this is how the 5 base buffer being utilized. So the total reaction volume I am considering 20 microliter. You can utilize it in 10 microliter as well. So primer should be not more than 3 to 5 picomolar. So how much primer you are utilizing for your cycle sequencing? Again 10 micromolar for most of your laboratory. Avoid. 10 micromolar is not a requirement for cycle sequencing because you are utilizing a linear amplification. It is not a logarithmic amplification you are doing in your PCR. That's why more primer is required for your PCR. For your cycle sequencing you require less primer because it is not a logarithmic amplification. Okay, it is a linear amplification. Don't use more than 5 picomolar, and our recommendation is 3 to 3.2 picomolar. Okay. So then BDT, either you can replace all 8 microliter in 20 microliter, so that is the 2.5x, or you can able to dilute it to 4 microliter or 1 microliter and add accordingly the buffers inside your tubes, so that you can able to go through your cycle sequencing along with the 5x sequencing buffer. So this is how the sequencing reaction being set up. So ready reaction mix, 2.5x concentration, BigDye sequencing buffer as per requirement with the dilution. Primer not more than 3.2 picomolar. Template, I already told the template quantity that you need to utilize for your sequencing, and the total volume will be 20 or 10 microliter according to your protocol. So sequencing reaction setup, so you will see that there is a denaturation step, and after that there is a hybridization or the annealing step. So now this annealing step 50 degree is not a reversal. It depends on your own primer. So if you are using a variable block PCR like Verity or any other PCR machine having the different temperature setup, so suppose 5 of your primers you are utilizing at the same day. So 5 primers can be set up with a different annealing temperature in the same PCR. Okay. So
one thing I have observed here is most of the labs are providing the annealing temperature is normally 50 to 55. Some of the cases it is 58.6 or 58.7. Why we do normally for 58.6, so it's set into 60 degree, the elongation temperature as annealing, considering as an annealing. Is it okay, or if I
you can keep 58, no problem. 58.5. Another this 5 seconds you keep it at 58.5. So that will be giving you accurate annealing of your primers. So PCR instrument always follow the protocols that is established in the PCR machine, either Verity, Proplex or Simpliam. Avoid annealing lower than 50 degree. If primer TM is greater than 60, eliminate the hybridization step. This step you eliminate if it is greater than 60 degree. Don't require. In case number of cycles of your signal is too weak or template amount is limited, so you can increase the number of cycles. Okay. So or decrease the number of cycles accordingly. And see the protocol for different variations as well. So then sequencing reaction cleanup. So we are getting the di-labeled extension products, unincorporated ddNTPs, unlabeled ddNTPs, and tris chloride, magnesium, polymerase, everything is there in the sequencing reaction. So the reaction consumes only a small portion of the di-labeled terminators present in the reaction. So the unused fluorescent terminator co-migrate with the DNA sequence, and you will get a very sharp peak. So at the sequence around 75 to 80 base pair, and also at the around 120 base pair, you will get a very sharp peak if it is not cleaned properly. So that is the dye block peaks or the ddNTP peak, that is aggregated. Okay. So this is the residual dye level terminators that is present in your sequencing data. So the purification, desalting or salt interferes with the electrophoretic injection and eliminates the remaining labeled ddNTP, and it is the labeled extension product that you are actually after cleanup you are getting this, and this is the unincorporated dye terminators that you are removing out of your sequencing products. So this is how the dye blobs looks like. This is actually the camera saturation data from the X2. This is not the sequencing data, this is the camera saturation data. So this is how the dye blobs actually looks like. So peak 1 to 8 is the dye blobs. So these are all the dye blobs. So these are the unincorporated BigDye Terminator dyes. So this is how the diagrams actually looks like. If it is a very huge diagram, you see that how much diagram is coming. So now diagram interfere with the base calling also. So if you have a smaller amount of diagram also or moderate amount of diagram also, you see that you know the base calling. Base calling is the quality value of your DNA calling. That means the if it is a peak and if it is the color, so then the software calls it as a base. Right? So based on the base calling probability, the quality of that particular base is actually recognized. And QV value above 20 is considered as a good quality DNA. Okay.
So QV value 20 means one error in 100 cases.
No, not one error. It is 0.1 thousand cases. QV.
No.
And QV 30 is
30 is
10,000.
1001 and 40 is 10,001. That's my
Yes,
yeah.
So it is the QV20 and when the bar height is high, so that means the QV value is more, like 30, 40, 50, 60 and so on. So that you will be getting a very good quality data. And in standard sequencing, the QV value mostly comes around 40, 45, 50, not less than that. So you see when there is a diagram coming, so there is a QV value less than 20. Less than 20 is yellow and red. Yellow is between 15 to 20 and red is below 50. So you are getting a very less quality of the data. Okay. So that you need to avoid, that you need to remove the diagrams. And there is a no block, you are getting a very good quality of the data. So purification method again, this is the post-cycle sequencing purification. This is not a PCR purification. Two purification are required. So ethanol precipitation, that many labs follow with the ethanol precipitation. Ethanol precipitation is good. There is no cost incurring with ethanol precipitation. But it takes longer time because you need to, after getting the cycle sequencing, you need to add your absolute ethanol, then keep it at room temperature for 15 to 20 minutes, incubate it, then centrifuge it for 30 to 40 minutes, then you decant it or aspirate it, so that at that time of decanting or aspiration, you will use some PCR product also. And after that, you again add 70% ethanol, the vortex it, then centrifuge it, decant it. So altogether it takes around 3 hours to complete. 2.5 hours to 3 hours to complete the ethanol precipitation method with the multiple steps. Okay. So now coming to the magnetic bead-based method. Magnetic bead-based method is also based on the ethanol precipitation, but it utilizes magnet that capture the DNA, like magnetic bead-based purification for DNA. Okay. So these are the polystyrene magnets which actually attract the DNA and remove out all the products in the alcohol. So in the magnet, you add the product along with the alcohol so that all the products are captured to the magnet. So then you aspirate it, add 70% ethanol, wash it, aspirate it, add high dye and take it out. So it takes roughly 10 to 15 minutes to complete the magnetic bead-based method with the alcohol. So then gel filtration. Gel filtration is tedious and costly, very tedious method. So because you need to filter to the gel, nobody utilizes into this gel. Membrane filtration, that also people do not use. And also there is another technique called the BigDye Xterminator. Xterminator is a proprietary from Thermophilia Scientific. So it is BigDye, you see the name, it is the Xterminator method. It contains the two solutions, one is the base and the SAM solution. Okay, so these actually you can able to add to your cycle sequencing product, vortex it for 30 minutes in vortexer, centrifuge it and run it with the machine. It takes total 30 minutes to complete with the 3 steps. And vortex, centrifuge, run. That's it. So one single pipetting and no reduction in your PCR products. So ethanol precipitation advantage is it is easy and cheap, reproducible and any format like single tube or 96 to 384 when you can able to do. But it is relatively longer method and special centrifuge is required to centrifuge out because it requires very high speed centrifugation. And use the tips, good tips is use 95% ethanol instead of 100%, especially when preparing 70% wash solution. Use protocol optimized for the BigDye Terminator chemistry. And sodium acetate for improving the recovery of short fragments if it is required. Use freshly prepared 70% ethanol. Always utilize the 70% ethanol freshly. And when you are opening the alcohol bottle for the first time, so when you just receive it and opening the bottle, after taking it out some alcohol, thoroughly tight it, and with some tapes or something you just wrap it. Okay, so that it will not evaporate or it will not absorb the moisture. So that is important. And better you keep that alcohol in 4 degrees so that it will be stable. Okay, that particular water you are utilizing for the cycle. And always prepare the 70% ethanol same day. Not like yesterday I have prepared, kept it in room temperature, not like that. Same day you prepare the 70% ethanol. It is true for any DNA positive, not about second sequence. So magnetic bead based method easy and reproducible, and automation is also possible, and it is not so very expensive compared to the gel filtration or other technology, but yes, it is expensive in terms of ethanol precipitation. Ethanol precipitation you don't require any cost, it is only ethanol and some EPD, no cost. So we have the dynamic sequencing cleanup kit available from Thermofisher. You can utilize that one. I am not going much about this kit, but it utilizes the dynamics and remove the unincorporated dye along with the salts and other DNAs, and then wash it with the 70% ethanol and dilute it with the particular hydride formula. So Xterminator. Yes, I will be taking half an hour.
Better to do troubleshoot.
Correct. Troubleshooting is next part is coming.
These things we are going to use.
Yeah, correct. So the Xterminator is also there. Xterminator is SAM and Xterminator solution you are utilizing. And I am not going much about the benefits and all. But yes, this is the fourth step I told you: add variant, seal plate, vortex, and quick speed and analyzing the machine. And this is the total data. You can be able to stabilize this solution for up to 48 hours in room temperature also, and it prevent the co-injection of the nitrogen extension products. And ethanol, you have seen that you add centrifuge empty plate about 2 hours it will actually take, and it takes about 35 minutes. And this is the data about with the BigDye Xterminator precipitation data. So now coming to the electrophoresis, I know that you know about the electrophoresis, and I am not going much about the electrophoresis and the capillary electrophoresis method. So mainly 50 centimeter capillaries are being used for all of your sequencing in your laboratory, and with the POP7 you are replacing the same. And there is a detection cell inside the machine on which the detections are done through the machine, and you need to maintain it properly. So do not touch the detection cell while you are opening the detection cell and all. So this is for the central facility, you need to actually take care of it. So run cycle, overheating, capillary filled with the fresh polymer, pre-run electrokinetic injection, voltage and data collection. So this is the cycle of run that actually occurs inside the machine. And these are the buffers and polymer utilized. And these are the polymers POP4, POP6, POP7, but mostly POP7 you are utilizing in your machine. And this is the machine you are utilizing, and today's date, that is the 6 Studio Flex that is newly installed in your machine laboratory. And this is the inside of your 6 Studio Flex instrument, and this is how the raw data converted to the sequencing data. So now I will be coming out mostly about the troubleshooting. So you have the array of softwares available also. So many softwares came along with your instrument, and many softwares are available online. You can able to utilize and leverage out of those softwares. I have discussed mostly about in my last presentation. So next, I will be taking 15 to 20 minutes about the troubleshooting part of your DNA data, that how you should actually check the troubleshooting data. But my suggestions will be, there is a DNA guidebook, sequencing guidebook available in our website. You just type out standard sequencing guidebook. Okay, this is around 250 pages of sequencing guidebook available, and you can download it free of cost from our website. And in that guidebook, all the troubleshootings are given with the picture, that if the sequencing data looks like this, what might be the possibility of the failure, or what might be the possibility of this kind of data. Okay, so all those troubleshootings you are able to get, like 60-70 types of troubleshooting, it is mentioned in the guidebook. Okay, today I am just taking some examples of the data and I will be describing it. So already I told you the template, primer, chemistry. So these are the different sources of the artifacts comes in your data. So this is the always run controls and running with a regular basis. for each of your sequencing reaction and troubleshoot samples with the positive controls and sample run on the 6-video French genetic analyzer with the POP7 or POP6 chemistry and analyze with the KB-based color. So there is a sequencing analysis software already installed in your machine in the laboratory. So if you are not having this software in your laboratory, come in the central facility and analyze one time with that software, because that software will give you all those quality value and everything together. In one shot you will be getting the data and transfer it to your pen drive or CD drive and come back to your laboratory for your faster sequence. So check the data quality and recognize the problem, that quality value of the electrophoregram, background signal, intensity of the signal, average signal intensity, and EPT view, all those you can able to troubleshoot. So first example is the electrophoregram. So that is, you are getting an analysis report of sample score of 11, LOR is 0 and average signal is 30. So that means there is a very less amount of signal available in your data. So now we are actually identifying the problems. Phenomena. So in the last slide I told you phenomena, cause and solution. So what is the phenomena? No sequence but dye blocks are present. So cause, no extension product formed during the sequencing reaction. So solution, optimize the sequencing reaction, that the sequencing reaction may not be positive or it is failed actually. So identify the DNA template quantity and the primer quantity quality to resolve this issue, because ultimately you need to amplify. If it is not amplified, then you will not get the product. Okay. So in your electrophoregram data, you might get this kind of data, but you go with the raw data. So suppose you are using a software which cannot able to visualize the raw data, you cannot identify this problem. That's why come to this software, sequencing analysis software, where you can able to go through the raw data. Raw data is very important. Next, sample score is 32, LOR is 779, that means you are getting a very good read length, and average signal is 67, that means signal to noise ratio is 0. So now here, what happens? That dye blocks are sequencing present, so you see these are the dye blocks. Suboptimal purification, and optimize the purification method. Complete removal of the supernatant is required if you are doing with the ethanol precipitation. Properly remove the ethanol, otherwise you will actually affect with the dye blocks. How many of you are doing your own cycle sequencing purification in your laboratory? No, it is done in the center. But dye blocks are present if it is not removed properly. So that is the main source of the dye block. So if you see any dye blocks in your data, come back to lab and check that proper protocol is being followed. So dye blocks should not be there in your data. So completely free, so that raw data, sample score is 32, LOR is 251. That means you are getting a very good data at the beginning, and after that you are losing the data. 251 means only 250 base pair sequence you are getting. Okay, and signal to noise ratio is 60. So gradual drop in signal intensity resulting in reduced read length. So the cause may be too much of DNA in sequencing reaction. That means you are providing very high amount of DNA that is not required. So but that is present in your sequencing reaction. So that also affect your DNA data. So optimize the amount of template DNA sequence, plasmid 150 to 300 and PCR product not more than 50 nanometers. If it is below 500 base pair, try to use 20 nanometers, not more. More TLM is this kind of data you may see. Then LOR is 552, sample score is 24 and signal to noise ratio is 792. That means signal to noise ratio is very high. So high background with the spatial pattern sequence cause presence of n-1 primer artifact. I told about n-1 or n+1 artifact. So this is how the data looks like. Very high signal to noise ratio in your data. So use primer without n-1 artifact and sequence complementary strand also with the other primer to get rid of the sequence. So last time I have checked one data that two primer being used, one forward and one reverse. Forward primer works properly, but reverse primer there is a pose modification. Or vice versa, I don't remember, either reverse or forward. In same sample, one is used with the forward and one is used with the reverse. One provided you good data and one is not providing you the good. It may be your primer also. You check. Okay. So then, here you see that LOR is only 22 and signal to noise ratio is very, very high. So, high background in entire sequence, it may be pull-up peaks, high signal noise value or rate spacing value. So, it may be because of the saturation of the camera, that means very high amount of sequencing product produced that saturates your camera. Or you can able to dilute the sample and then re-inject it with the reaction. So, you need to check it if you are getting a very high data and also the noise, the signal to noise ratio is very high. So, PCR related issue, I told you about this salt. If there is a salt, so then short LOR you will be getting. Template has residual high salt concentration and too much salt from PCR from template could reduce the tag polymerase activity, and that's why you are not getting a sequencing data after 100 or 200 minutes. PCR related multiple product. So you see that you are getting a PCR product but the quality value is not good. So it may be because of another peaks are coming. It may be a non-specific binding of the primer to the template during the PCR of cycle sequencing. So check the primer and optimize the PCR. Residual PCR primer in the cycle sequencing mix. It may be non-specific PCR. It may be residual primer or the primer dimer. Multiple clones or colonies present during the sample preparation of the PCR product. And contamination from water or environment and reusing septa. Septa reuse. The septa you are using, so it is not properly cleaned and reused. That also may give you another.
They are not using it for us.
No. Yes. I am giving you all the scenario. That's what. So when we are getting some trouble, we need to identify the trouble. So that is the main thing. So if you are get rid of the trouble, so you will not get it everything.
So as you told, the cleaning process of septa is normal warm water.
Warm water, dry.
Previously they have used hypochlorite solution also.
Actually, ideally you should not reuse the septa because using the septa can contaminate also. Chances are there.
Better to not.
Okay, better to do not reuse. One or two times cleaning, you give the good data is okay. But if you are filling with the
data. What about the septa is? They know many, but still one.
Septa is basically, you know, for any PCR, you are covering with some film or something. But in sequencing, there is a capillary. So when the capillary will be going inside the plate, if there is a film, it cannot able to go. So septa is a one-directional valve. It is a rubber bed and valve is inside, so that the capillary will be going here. So when the capillary will be go, then it will open. Otherwise it is closed. Just like that. So it is called septa. You go to lab, check what is septa. Then it will be visualized to you. Okay. There is a rubber bed that is actually used on your plates.
So, in the previous one, so how the paper which has like N-1 amplified look different?
No, it is not N.
Yeah, it is not that, but how it looks different?
So, N-1 artifact will be, for every sequence, there will be a low noise in the PCR. So here also low noise is there, but here the product height will be high. Because in N-1, what will happen? Some of the fraction of the primers got degraded. So that will get a lesser noise. So you concentrate on the signal to noise ratio. That's why I concentrated that what is the signal to noise ratio in your data. If the signal to noise ratio is high and also there is a peak under peak is coming, so then it might not be your N-1 problem. It is another product. If it is another product, so then the peak height will be high. So now, in a good quality data also, another product may come. Can you able to explain to me? Means there is no problem. These troubles are not there in your PCR product. Your quality of the PCR product good, your sequencing is good, but you are observing, suppose you are getting 50 base pair data, and after that there is a two noises coming. One after one, one after one.
Yeah, my experience is it's in there if it is insert deletion something is there.
Yes. So it is indel. So when there is an insertion deletion in your DNA, that means one insertion and one deletion in your product is there. So because of indels, two products will be coming. So that's why you will be getting two bases together.
Yeah, lengthwise differ.
Yeah. So it is not a problem in your data. Go with another primer and check that the same position that noise is coming from. If it is, then you report it as the indel. Indel data cannot be analyzed with the Sanger sequencing. Okay. So then the peak under peaks. So these are the peak under peaks. So peak under peaks is the pattern that actually provided the peaks are off scale, and similar pattern occur if the wrong spectral matrix is used, or the spectral matrix is expired, or the capillary is being not proper in good condition. So then also you will be getting this phenomenon of pull-up peaks. Pull-up means another peaks are coming. So the dyes are getting pulled up. There are 4 dyes. So 4 dyes will there maybe the crosstalk between the dyes. So to reduce the crosstalk, we are utilizing the matrix for calibration, so that the pull-ups will be reduced. How come? দুটো কালার আছে, একটা ব্লু আছে। Suppose this is one blue spectrum, and this is yellow spectrum. Okay. So now, when we are calibrating, so we are getting a blue spectra over here and one yellow spectra over here, peaks over here like that. Okay, because we are getting these peaks only. We are avoiding these peaks at the time of calibration. So now see that this also there and this also there. So that means we will be getting another peaks over here and you will be getting another peaks over here. So this is not your DNA, this is the pull-ups of that particular spectrum. This is the pull-up of that particular spectrum. So this is nullified by spectral calibration of your system and the capillary. So now if the capillary is older, so it is used many times or any other things happens, so then that pull-ups will come more. And if that pull-ups are coming, so then you will be getting this kind of data. Pull-ups peaks. Pull-up peaks are not like that the previous one I have shown you. So this is the peak under peak. Peak under peak and high peaks. So pull-up peaks, you see that it is not exactly coming here.
Extension of the previous peak.
Extension of a previous peak also. So if the pull-up peaks are coming, so then you change your capillary and recalibrate your machine with the proper capillary and capillary.
Okay, in that case, without changing capillary, is it possible to only do spectral?
It is very rare. Means I haven't seen many times this kind of issues. So pull-ups may be coming when the capillaries are not properly maintained and it is used for like 300, 400, 500 reactions. So then it can happen in the capillary. So now example of primer timer. So it is the forward and reverse primer. So high background noise at the beginning of the sequence and possible primer timer coming from the PCR reaction and optimize the PCR primer removal from the PCR state. So from the reverse and forward you are getting around 50-60 base pair of the noisy sequence. It is the primer timer. Tailing peaks, possible array replacement. So if there is a tailing of the peaks like that, so this is the tailing of the peaks. So in all 4 colors, usually the first sign indicates array is starting to wear out and need to be replaced. So this is because of the tailing peaks. So some peaks for GC and GT and G regions use 1 to 2 molar betaine in PCR and cycle sequencing, 5% DMSO in PCR, but in today's date people do not change in their PCR reactions.
So what are trailing peaks?
Trailing peaks means if you are getting this kind of peaks, so peak should be like that, na? So the sequencing peak should be like that. Okay, but tailing peaks will be here like that. This is the tail. Tailing means the extra peaks are coming here. It is not like the uniform peak. So this is the tail. This is the tail of the peak. This is the tail of the peak. Okay. If these tailing peaks are coming from every peak, so that means there is a wear and tear in your capillary area, and you need to replace your capillary area. So these are all the things that you need to follow for your GC and GT sequences. But in today's date, I haven't seen that people are using a different concentration of this or DMS in their reactions. So successful sequencing of GC sequencing. So this is the Terminator 1.1, and this is the dNTP BigDye Terminator sequencing kit. So in that case, high GC rate, we do have a separate kit called the dNTP BigDye Terminator kit that can be actually solved with the high GC rate sequencing as well. So other modification, initial denaturation of the DNA templates, example 5 minutes at 98 dB. Prior to adding sequencing chemistry, increase the number of cycles if signal is too weak. For large DNA template like the backneat, cosmic or bacterial genomic DNA, initial denaturation step of the template, you can able to run a cycle at 95 degree and then the annealing and extension at 60 degree and so. So 80 weeks template, reduce the extension temperature protocol, reverse cycle sequencing protocol and template with secondary structure like the similar approach as in GC sequence you can able to approach for. Modification of the cycle sequencing protocol, I already told that if there is a high amount of sequence or low amount of sequence, accordingly you can able to increase or decrease the number of cycles. So from 25 cycles you may increase 1 or 2 cycles to get the more amount of product. That may happen actually. Thermocycling protocol recommendation. So add initial denaturation at 5 minutes to 96 to 98 dB and increase the number of cycle sequencing if signal is too weak. So this is the purification problem or time blocks I told, raw data and analyzed data, and use the correct protocol, taking it already I told you. And with that, today I am actually thanking you. And one more thing I would like to show you that I have shared with some tips and tricks with the laboratory. So this is what I have presented in my presentation. So this is as a whole, I have prepared this short PDF document that how you could able to get improved quality of the DNA sequence, examining the DNA template quality, quantity, whatever I told in my today's presentation. So that DNA quantity, time and design, then the primer, quantification, storage, calculation of 100 micromolar, contamination, what should be avoided, minimize the contamination. So then contamination that affects sequencing, sequencing reaction setup, reagent handling and storage, and the sequencing run module. Everything I have provided with the 5-6 pages, so you can get it from lab and take a printout or in your laboratory and follow those protocols as well.
It is with us.
So with that, I think that you improve your DNA sequencing products and come back to us. So we, our application teams are there to come back to us and let us know that what kind of problem you are facing. I don't think that problem will persist. You will get a very good quality of the data. And anything you will get. Thank you. So any questions?
One thing is left, that is the jail picture you were.
Yes, please show me so you can connect
that to the
your
laptop.
laptop.
So that. Please go to there and go with me. So that.
So these are the two PCR sequences that I had given for the sampling, sequencing samples on 11. So,
What is the result of the year?
What is
What was the
what is
yeah,
the
result I will show you the result also. So this is the clean band after elution. Even before elution it was a single band. That picture I don't have because I was running an elution there. Okay.
But you need to show the picture before elution. So that is very important.
Okay, that usually we don't take the picture there. We run that white foam elutions and we load the samples and we then extract it. But yeah, I have it. I have a single band from it. That much I can. I don't have the
Why
picture.
I'm telling it because that actually
Gives the idea
gives
that
you the scenario that what should be your troubleshooting of your data because it is already gel extracted.
Okay.
So there is no primer dimer, there is nothing, or some training on DNA or anything is there. So that you need to identify.
So sir, this is from one of the sequences at 925 to 1249, one sequence that I had given for sequencing. And the other that...
So you got the entire sequence out of that product?
No, actually I left first around 70-80 base pairs.
So is there any dialog kill?
No, nothing.
No.
Dialogues were not there,
Mhm.
but there were very low signals, so I left that
Up
part.
to 50 base pair?
Yeah,
there is
so
a-
around 80 I left. I took it from 90 base pair to 300.
Hmm.
The sequence was good over there, so I took that.
Why 300? Because it is
After 300, the signal was dying off.
Okay, so is it done with the 1 hour protocol, fast protocol?
That I have to check because I don't know.
If it is fast protocol, I told you that way that you can rerun with the 2 hour protocol so that they can get a higher.
Maybe the first is gives you little bit of it.
Okay, and so this is that for another sample. The PCR band was there, 601, though the labeling is wrong, actually, the 109 is given. But this actually I got a signal like this. There is no signal, but however there is DNA and the concentration is around 100 nanogram when I
So
checked it on
maybe it is a reverse primer or the other product.
other product.
Other
Other product.
So there may be some issue with the binding of your primer and all that is not giving your product.
Okay.
And also I told you in gel extraction, so
There is
because
a loss of,
there is a loss of.
Okay, so after elution I had got around 100
So
nanograms.
my suggestion will be that avoid gel extraction.
ExoSap method would be
ExoSap
better.
method
Actually, in
will
our
be better.
lab we don't do it. We usually do the gel extraction
Because it
method.
ultimately takes your own time of gel
Yeah, that
extraction.
is there.
Your cost and also the
Actually, we
longer protocol
run the
as
gel
well.
and then we purify it on the Kaizen columns.
Hmm.
So, and then we heat inactivate. So, all those 3 points you mentioned, actually we follow them while the purification step, except for the enzymatic processing, we do the other.
If you have this PCR product available with you or you are loaded
I
all
had
the-
loaded the entire
all
thing. I had to
entire-
set up a little bit and
So then you set up another PCR and go with the ExoSap and
I
check
have to
that.
find where I can get ExoSap. That thing I
Yeah,
want
so
to.
one thing I want to
ExoSap is
mention, so one thing I want to mention that, so if you are doing the column thing, what is the cost of rough one single column for purification part?
That
Estimate to approx.
That I have to find out what's
Okay,
the
because in the
just.
ExoSap, I think 25 to 30 per sample, right? It's little bit of money. It is.
Yeah, it should be cost effective. The ExoSap should be cost effective.
And the time, less time.
Very less. This actually requires the gel itself runs for around 45 minutes to 1 hour.
You don't know.
And
maybe
after
maybe,
that, we have to go for the extraction. So it
Gel
is
running,
Okay,
cutting
another
and
thing
all.
I asked to Mr. Sunil, suppose they did the gel purification thing. So will I need to do the ExoSap again in our center visit? Not required, right?
If they are telling you it's gel extraction, go directly
Only
to
when
the
they are saying that it's a raw PCR product, then only you do ExoSap,
So
right?
you prepare a one-page document.
Document, yeah,
So you,
submission form.
submission form. You
Absolutely,
ask
that you need to
the
modify,
PCR product, if it is purified, which method of purification,
Yeah.
gel picture. So all those documents
Things should
should
be
be
clear.
that should
Yeah.
be provided along with the PCR product
Yes.
or the plasmid DNA. Okay.
Fine.
So that you can identify. If it is
So-
already purified,
So
there is
no need to, yeah, no,
uh,
suppose double time purification is
no,
not
no
needed.
requirement.
Also, one more thing that I just checked with the Kaizen extraction kit that the EB, that is the elution buffer that we use, it's a TRICDTA, that is a 1 millimolar EBTA buffer. That it's on the low EB
It should
buffer.
be low EDTA.
That we don't use for extraction, for gel extraction or for plasmid purification, whatever
All
it
is the normal
All
thing.
is that same
Yeah,
elution
so
buffer that
one
we use.
thing for the good thing is good for you and for others.
Yeah.
But my focus is to bad thing, right? The fail means whatever. What is your assumption, anticipation of regarding the second one?
Means another product, it is.
So can you show that
so,
data?
so when we were doing the PCR, so it is the, as I told you, that it is a
Forward and reverse.
forward and reverse primer, both you are providing and it is doing the PCR.
Yeah.
So now when you are coming up with the cycle sequencing, it is
Only one prime.
primer.
Yeah.
And also that annealing temperature and all you are setting up. So that may be something different than the annealing temperature that they are setting up for the PCR. So it is because of the same that the primer is not binding in that side. So that need to be troubleshooted.
see, the PCR product is more accurate product length, right? So the chances are very high to attach reverse primer to the pin. So,
here I could see that from this data I couldn't able to see the raw data because this is not the software. Biologist cannot handle the raw data, but we can see in the software available in the machine that you can also check the raw data.
Yeah.
So from the raw data you can identify that what is the peak height scale. So if there is peak heights but there is noise, so that means some other products are there. But if there is no peak heights, that means PCR is
no, can you do illumination of that thing so that because the bigger peak, that's why maybe lower their peak notch is not.
No, here in this software,
extend that peak,
the x-axis will not
yeah,
take it.
x-axis, yeah. Possible, but they are up. No.
X-axis.
Yeah, that one up. Y-axis, not x-axis. Why? No.
No, that zoom is only possible. I don't think.
Okay.
Yeah.
See, there is some product, but there is some other product is also coming.
Because of the bigger peak, the peak
It is actually the other product that is came along with the product. That's what I could see, but I need to check the raw data.
It's a kind of mix.
If the raw data you can tell me, then I can
Yeah,
check.
I can tell the sample number.
You can come in the lab, I can check the data also.
Yeah, the sample number will be
Yes.
available. And so another thing, the data that I got, like the other sample, that I actually got a signal for from 90 to 300. Over there, I did a sequence matching with the multiple sequence alignment that we normally do, that whatever sequence I'm getting from the sequencer and the original expected sequence. There were a few mismatches. So I couldn't get the expected sequence either. So that was there. So there might be a problem with the
The
sample.
quality of each of the bases came correctly. So
It looks somewhat like this. Okay, so yeah. So there are breaks in the matching.
There is not exact matching.
Not exact matching as it should have come.
Yeah.
So basically the data I have given it previously for sequencing in the previous facility that we have, and we had got very good sequence, like complete matches. Region of it, not the entire region, but whatever sequence was there, it was intact given in the.
My side will be that you just redo this 2 PCR and send it to the laboratory, and they will be doing it with the particular exam level check. And what I could see that I do not know why the band are so thickened, even for the DNA marker.
Yeah, the DNA marker has run.
Marker and also the DNA bands, it is not the sharp band.
Okay.
It is actually spread out. So can you tell me the reason why it is spread out? It is a reused gel or something?
No. It's a fresh gel.
It should not be spread out.
Like
Even
the
the ladder also spread it out. So it should not be.
Is that maybe a problem with the running of the gel or the sample loading? Yeah. But yeah, I can rerun the gel, of course. When I'm doing the PCR once again, I can run
No,
another
better
gel.
now you give us the only PCR.
Only PCR product. Then I
Yeah,
can
you
do
only
that.
PCR products and all you take the actual gel picture of the
Before
PCR.
elution.
Before elution. Okay. Before elution.
You don't need to do that, you know.
And you don't perform any gel extraction.
Okay.
You give it directly.
Let's see what is.
Okay, so that I can do. It will take around 3 hours for this PCR to run. So
Well, but tomorrow morning you give it
yeah, that
to
should be
us.
fine. I can set up a PCR and hand it over to you.
Anyone you have
So, sir, with everything run as expected, so how much could we use?
up to 1100 base pair with the 2-hour run protocol. That is a standard run protocol. If you suppose she is having 972 base pair.
Yeah.
So 972 complete you can get from one sequence, one day. Correctly you will get. So if your product is good and if you want a 900 base pair sequence, don't dilute more than like 1/4 of the sequencing reaction, because when you are diluting the sequencing reaction, the dNTP enzyme, everything will be diluted. Okay, if you are taking 1 microliter and expecting 1000 base pair, you will not get it.
And sir, one more thing, if we are when we are giving only the PCR product to you, like this time I will be handing it over, that will be in whatever enzyme mixture it's going. It's going
Yes,
completely.
yes.
That is mainly
They-
NFW. That's it.
Actually, after the PCR product you are submitting to the lab, they will take 1 or 2 microliter,
Yeah.
because the PCR product concentration should
Already,
be good.
yeah, already
It should
it's
be
high.
like 50, 60 nanogram per microliter, so that they will take 1 microliter
Yeah.
and add 1 microliter of water.
Yes.
That.
That will
And then they will go for the ExoSap.
Sequence.
ExoSap. And
ExoSap
then
and
after
then
the
the-
ExoSap, they will be going for the sequence.
And for if in case we need to do the gel extraction, in case we need to do it, then is it advisable to dissolve it in NFW? Why? Because this is a short-term period. Facility.
You can do it in NFW, but NFW should be of good
No, that's a good
quality.
point. He can activate good maintained aliquot of NFW. Then will that give
It should not be from your your source of milling or something. It should
No,
be
it is a package, DPC treated water. That is what we use in our lab.
What we have seen, why I am telling you, from every laboratory, suppose here in Nadia, you are getting a soil pH of 6.5. So actually, whatever the milky water you are getting, it will not meet 7 pH. And from suppose Kolkata, it is more alkaline, you will get a 8 pH water over there. So even if you are actually purifying it with the mildew system or some system, you will not get an ideal pH of the water. And we have seen the substantial changes in the pH of the water in different regions of the.
No, but this is what we use as a proper package.
Package HSP water you utilize so that they maintain at least the pH.
It's a fresh aliquot and then it's heat inactivated and then it's used for
the samples. Let's see.
Okay.
See, all these things are there.
We should standardize before we can.
Yeah, so all these things are there. So one single plan might happen that fail. Might
Means
happen.
recently, just 1 month back, we have done another training at Badoni. They are also having a center facility for sequencing. They have given almost 70 PCR products for sequencing for the training purposes from different laboratories. 1 or 2 failures.
That
So,
can be the.
so that why I am telling you that it is so robust that failing will be less possibility. So even I am telling my experience in my PhD days also. So I used to do the Sanger sequencing for the samples from the patients. Okay, it is a human patient sample. We used to collect the blood or tissue samples, extract the DNA and go for the Sanger sequencing. So my guide used to tell me for each primer you show me forward reverse sequence for at least 3 sequence first. If that sequence does not fail, come back, then you take most of the samples for same primer serial for the sequencing. So because I am supposed to do population studies for the so that is my experience. I am telling you that 1 or 2 will indicate you whether it is failing or not. If it is not failing 1 or 2, that means it will not fail for rest of the samples. So that's what you need to establish.
Okay, so is there any questions or we are close? Okay, so thank you first of all,
Thank you
thanks
so
to you
much.
for your coming to here for giving them an idea about the thing. And you, so what we'll do in our end, for the everything we will make a form, modified thing, and we'll circulate to all of you so that you can understand what are the QC things by taking all these checks and things. And accordingly, you provide your. After that, it might happen that one of your data gets here, so we'll sort it out. Yeah, we have to minimize the error. So 90, we'll try to do it 90% or 95%. If not, like initially may not be. So it may be 80%. We'll try to increase it. Okay, with the help of you. Okay, you guide us what to do and what to do.
One thing it would be helpful if you could circulate that PDF.
Which one?
That tips and tricks.
Yeah, obviously.
Is
The 2 mail ID is fine, so that everybody will get, right? One is research student, the CBS research student, everybody go.
Yeah.
I'll send it. Whatever he has sent, I'll directly say no.
So, I would like to contact me like so that
you can write it down.
Thank you all for coming here.
So my
Okay, by today, evening, we'll take the samples. If not, tomorrow morning, you will. But try to be at 11 o'clock by tomorrow.
So this is my
Today evening, you are
contact
at
number.
5. You can leave samples.
My mail ID is Sanjeev, SANJIV @thermofisher.com.
Yeah, so that immediately.